Hello students, welcome to EPG Padasala. I am Dr. P. Nagaraj, Assistant Professor from Anna University, Chennai. Today we are going to talk on module carbohydrates as a structural backbone from the paper Structure and Function of Biomolecules. Objective of today's lecture is basics of carbohydrates, monosaccharides and its properties, oligosaccharides and its properties, polysaccharides and its structure, examples of some of the polysaccharides and application of polysaccharides, of course, glycoproteins and its function. The definition of carbohydrates, which is monosaccharides, simple sugar, cannot be broken down into its simplest form under mild conditions. Oligosaccharides, a few or usually 2 to 10 monosaccharides combined together will give oligosaccharides. Polysaccharides are polymers of the simple sugar will give the polysaccharides. Glycosidic linkage. The glycosidic linkage is nothing but linkage between two glucose or galactose molecule between C1 to C4. Normally it may happen. Example, if the one monosaccharide unit linked via another monosaccharide via 1,4 linkage, it will give as a disaccharide. Either it may be in the alpha or beta, that depend upon the nature of the bond forming, that will lead to disaccharide. It is extended to trisaccharide via it will make up to polysaccharide. So glycosidic linkage is nothing between linkage between two sugar unit via ether linkage. We can also tell that it's 1,4 linkage, sometimes 1,6 linkage, etc. may happen. Disaccharide, of course, there are examples like maltose, cellobiose, sucrose, lactose. These are all the examples for disaccharide. In case of maltose, two sugar unit, namely glucose and glucose, which is linked via alpha linkage. 1,4 alpha linkage will give maltose. In case of cellobiose, again glucose 2 units linked via 1,4 beta linkage, that will give cellobiose. Same way sucrose, which will give D-glucose and D-fructose via alpha beta linkage, will produce sucrose, again is a disaccharide. In case of lactose, galactose and glucose, which is linked via beta 1,4 linkage. That alpha beta, that may be depend upon the one sugar unit, that alpha carbon will decide. If it is axial, that is alpha linkage. If it is equatorial, that is beta linkage. Higher oligosaccharides, that means oligosaccharide is the saccharides which is higher than disaccharide. That means that monosaccharide or monomer may be present more than 2 or maximum up to 10 or 11, that is comes under oligosaccharide. Here some of the examples are given, where you can see there is a trisaccharide, raffinose, racinose, lectyl, etc. are all belongs to this kind of oligosaccharides. While hydrolysis oligosaccharides, you will get more than 2 monosaccharide unit, that belongs to oligosaccharides. Slide 9 again, oligosaccharide groups are incorporated into many drug structure, which is shown here, especially oligosaccharides. 3 units of monosaccharide link via ether linkage, alpha or beta, that having the linkage with some medicinally active molecule, that will be having very complex chemical structure, which will lead the functional group tolerance as well as act as a medicine in most of the cases. That and all comes under this kind of oligosaccharide. Polysaccharides, the nomenclature of polysaccharides is, we can say broadly classify in 2 categories. One is homopolysaccharides as well as heteropolysaccharides. If you do the hydrolysis, you are getting same type of monosaccharides, which is called homopolysaccharides. If you do the hydrolysis, you are getting 2 different or more number of different types of monosaccharides, which is heteropolysaccharides. So starch and glycogens are storage molecules, dietins and cellulose are structural molecules, cell surface polysaccharides are recognition molecules. So these are all the different type of polysaccharides which is available in nature. Polysaccharides again is a kind of saccharides where more number of monomer unit which is linked via glycosidic linkage, and that is comes under many categories, especially primary structure is a linear or alternative which is present in the polysaccharides. That is monomer which is linked via glycosidic linkage may produce this kind of polysaccharides. Secondary and tertiary structure, it will deals with rotational freedom, because each sugar unit linked via O glycosidic linkage which is have free rotation, which is a single bond which can rotate freely, and also it is possible more number of hydrogen bonding, because the hydroxy group present in the carbohydrate molecule which will make a hydrogen bonding. Then oscillation of course which is possible. Local, secondary and overall tertiary random coil helical conformations possible in case of secondary as well as tertiary structure. Movement around bonds, that means the single bond which is linked between two monosaccharide unit, that is easily freely rotatable. So that is how the fluctuation or the molecule orientation can change depend upon the nature of the bulky molecule. In case of polysaccharide, the orientation of the molecule may be possible via linked glycosides. Why? Because of it is a single bond which can be easily go for rotations. So in case of tertiary structure, the size especially beta-1,4 linkage, zigzag ribbon like linkage will be there, whereas beta-1,3 or 1,4 alpha linkage you will give halo helix or U-turn link will be there. In case of beta-1,2 which is twisted or crumpled, whereas in case of 1-6 no ordered conformation. So these are all the conformation if in case of bulk tertiary structure will give, especially if more number of sugar unit linked together will produce this kind of shape. The shape of orientation of or geometrical conformation will change depend upon the monomers and the nature of monomers. That can be explained in this one. A ribbon type structure also possible, where each molecule orientation may involve, apart from the glycosidic bond, there might be possibility of making hydrogen bond in between the two units of sugar. So it will make overall, due to the hydrogen bond, the entire conformation will change. Like in case of cellulose, it will make flat ribbon type conformation. Chain can align and pack closely together, also get hydrogen bonding and interactive force. So this may lead another class of conformation change in case of cellulose molecules. Buckled ribbon type conformation, especially in case of alginate. Alginate is a kind of polysaccharide that is having different conformation because of the free rotation of the glycosidic bond as well as the hydrogen bonding. It will make different kind of conformation that is come under type of buckled ribbon type conformation. So another class of structure is halo helix type structure. Tight helix void can be filled by including molecules of appropriate size and shape. Based on the size and shape, it can be fixed into the helical structure. More extended helix, which is another class, 2 or 3 chains may twist around each other to form double or triple helix. That is maybe even the size and shape which will vary appropriately. And very extended helix, the chains can nest, that is close pack without twisting around each other. So this kind of helical structure may be possible. Based on that, the helical structure, it will do the biological action in our body. Amylose forms inclusion complexes with iodine, phenol, and N-butanol, etc. Because these kind of molecules, amylose can make a conformationally flexible molecule that can make bond with like other iodine or phenol or N-butanol. Because of the nature, it can orient themselves, reorganize themselves, it will make such a compound within itself. That kind of possibility is there in case of bulky molecules. A liganded amylose iodine complex as shown in the picture, the amylose unit surrounded by the iodine or complex rose iodine atoms neatly fit into the core of the amylose helix. So amylase unit which is surrounded by iodine molecule in between the amylase unit, which will make like the liganded amylose iodine complex, which can be shown in the picture. Tertiary structure again conformational zone, zone A, zone B, zone C, zone D and E. In case of zone A, extra rigid rod type of conformation available, whereas in case of zone B, rigid rod, example is xanthan, and in zone C, semi-flexible coil, example is pectin, and in case of zone D, random coil, dextran, pullulan, etc. And in case of zone E, highly branched amylopectin and glycogens. So this kind of zone wise it can be categorized based on that tertiary structure and conformation of complex carbohydrates. Quaternary structure of aggregation ordered structure. Aggregate and gel formations may involve other molecules such as calcium 2 plus or sucrose, other polysaccharides, mixed gels. So this kind of conformation may possible in case of quaternary structure. There are several polysaccharides we can describe as case studies, like number one is alginates, second one is pectin, and third one is xanthan, fourth one is galactomannans, and fifth one is cellulose, and last sixth one is starch. So these are all belongs to naturally occurring polysaccharides. The examples and details, properties, application, everything we can do one by one. The example of complex carbohydrates, one is alginate. The source of alginate, brown seaweeds and linear unbranched polymer containing beta-1,4 linked D-menuronic acids and alpha-1,4 linked D-glucuronic acids residues, which have link between these two. Not random copolymers but consist of blocks of either menuronic acids triple or glucuronic acid triple, or mix between these two will give alginate complex carbohydrates. The structure was explained by poly D-menurate. The mannose unit having carboxylic group at C6 will give you this complex structure. Similarly, poly L-glucuronate having the glucose unit having the acid group at C6 position will give the poly D-glucuronate. The structure may be very complex. More explained using this kind of structure, where in between hydrogen bond present between the two units will make conformationally more stable as well as flexible molecules. In case of all glucuronate will link via this kind of structure, whereas in case of menurionate doesn't form this kind of hydrogen bond. And in case of mixed between these two, like a glucuronate and menurionate, some of them having the hydrogen bond, some of them doesn't have hydrogen bond, the orientation will be totally differ compared to those exist in the previous. Also explain the aggregation of all the menurate and glucuronate will with the calcium atom in the middle. So calcium poly alpha L glucuronate left-handed helix view down axis, view along axis shown the hydrogen bonding and the calcium binding sites. So in case of vertical, if you look, you will not see the hydrogen bonding, whereas if you do the horizontal, you can see the hydrogen bonding present in the unit as well as bonding site in case of calcium 2 ions. Different types of alginates, different properties, example gel strength, polyglucuronate, gelatin through addition of calcium 2 plus ion, especially egg box, polymenurionate, less strong gels, interactions with the calcium 2 plus, weaker ribbon type conformations, alternating sequence, disordered structure, no gelatins. Properties and applications: high water absorption tendencies, a low viscosity emulsifiers and shear thinning thickness, and stabilize phase separations in low fat, fat substitute example as alginate, caseinate and blends in structured three-phase systems. Used in pet food chunks, onion rings, stuffles, olives and pie fillings, wound healing agents, printing industry. So these are all the applications which I have seen. Another class of complex carbohydrate, example for pectin, the cell wall polysaccharide in fruits and vegetables which is mainly available. Main sources is a citrus peel which is having more amount of pectin. The structure has shown their partial methylated poly alpha 1,4 D-galacturonic acid residues and hairy regions due to presence of alternating alpha 1 to L-raminosyl alpha 1,4 D-galactanosyl section containing branch point with side chains mainly L-arabinose and D-galactose. Properties and application of this one is a main use as a gelling agent, especially jams and jellies, dependent on degree of methylations, high methoxyl pectins gels through hydrogen bonding, and the presence of sugar and acids. Low methoxyl pectins gel in presence of calcium 2 plus, and also it is used as a thickeners and water binders and stabilizers, etc. Xanthan is another class of complex carbohydrates where beta 1,4 D-glucopyranosyl backbone with side chains of 3 to 1 alpha linked D-manopyranosis and 2 to 1 beta D-glucuronic acid and 4 to 1 beta D-manopyranose on alternating residues. This how the molecule will totally complex as well as branched chain as a straight chain will make the molecule more flexible in terms of making hydrogen bond as well as the single bond orientation. Properties and applications: double helical conformations, pseudoplastic, that means which is not a normal plastic, which is a plastic like material, shear thinnings and thickener, a stabilizer, some of the cases you may act as a stabilizers, and emulsifier most of the cases, and foaming agent, forms synergetic gels with galactomannan. So these are all the properties and applications of galactomannan. Another class of example is galactomannan, which is the structure is beta 1,4 mannose, backbone with alpha 1,6 galactose side chain. Ratio of M to G means mannose and galactose is varies. In case of 1:1 mannose and galactose will give fenugreek gum. It is mannose glucose ratio is 2:1, so guar gum. Similarly 3:1 will give tara gum, 4:1 give locust bean gum. So these are all, if the one unit varies, totally different product you will get. The structure of guar gum obtained from endosperm of Simnopsis tetragonobulbs, and the structure is the glucose as well as galactose which is linked via 1,4 as well as 1,6 linkage. Similarly locust bean gum, there is only one unit because out of 4 unit only 1 unit of glucose present, whereas in case of tar gum there are 3 unit of mannose and 1 unit of galactose present. Properties and applications, which is non-ionic in nature, solubility decreases with the decreasing galactose content, thickeners and viscosifiers, and used in sauce, ice creams. LBG can form very weak gels. So these are all the properties and applications. Cellulose is another class of polysaccharides where beta 1,4 glucopyranose is the repeating unit will make the polymer or carbohydrate chain. Properties and applications of cellulose. Cellulose found in plants as microfibrils, very large molecule, insoluble in aqueous and most other solvents. Flat ribbon type structure allows for very close packing and formation of intermolecular hydrogen bond. 2 crystalline forms, cellulose 1 and 2, derivatization increases solubility. Examples hydroxyl propyl methyl cellulose, carboxymethyl cellulose, etc. Starch, again which is along the naturally found carbohydrate, a plant storage polysaccharide, two forms of starch, one is amylose and another one is amylopectin. Most starch is 10 to 30% amylopectin and 70 to 90% amylopectin present. Average amylose chain length 100 to 1000 residues. Branch in amylopectin every 25 residues. Amylose has alpha 1,4 linkage, one residue end. Amylose and amylopectin. Amylose is a straight chain polymer where there should not be any branching. It's a linear polymer, whereas amylopectin, which is a linear as well as branched polymer, that we can represent by the structure. So which is a complex in nature, amylopectin, simplest nature in case of amylose. Starch is again a complex carbohydrate. Amylose is purely soluble in water but forms micellar suspensions, whereas in the suspensions amylose is helical in nature. Glycogen is another class of complex carbohydrate, a storage polysaccharide in animals. Glycogen constitutes up to 10% of liver mass and 1 to 2% of muscle mass. Glycogen is stored energy for the organism. Only difference from starch, number of branches, alpha 1,6 branches every 8 to 12 residues. Like amylopectin, glycogen gives a red-violet color with iodine. Glycogen structure, which is shown here, is a kind of branched carbohydrate where the main chain will be there as well as branched chain will be there. The main chain having the linkage between alpha 1,4 linkage between two glucose unit, whereas in case of one alpha 1,6 linkage also form to make the branched chain. Dextrans is another class of complex carbohydrate. If we change the main linkage between glucose from alpha 1,4 to alpha 1,6, you get a new family of polysaccharide, that is dextran. Branches can be 1, 2, 1, 3 or 1, 4. All the possible of branching is there. And apart from that, dextrans formed by bacteria are components of dental plaque. Cross-linked dextrans are used as Cefadax gels in column chromatography. These gels are up to 98% water. Dextran is again linked with monomers as a sugar, especially glucose, which is via alpha 1,6 linkage will give dextran. Cellulose, which is a naturally occurring polysaccharides. Cellulose is the most abundant natural polymer on earth. Cellulose is the principal strength and support of trees and plants. That is cellulose, which is made up of beta linked glycosides, that means 2 glucose unit linked via 1,4 beta linkage. It won't undergo easily hydrolysis. That is why all the plant material which is very hard because of the beta linkage between the two sugar. Cellulose can also be soft and fuzzy in cotton. So cotton material fully made up of cellulose, that we can go for fiber again, and tea barks and stem, everything which is made up of most of the cases in plants which is made up of cellulose. Cellulose having the 2 glucose unit, 1,4 alpha glycosidic linkage. Cellulose is another class of complex carbohydrate which is fibrous in nature, which is available most of them in naturally. Cellulose versus amylose. Amylose is alpha 1,4 linked D-glucose units. Cellulose again beta 1,4 linkage make all the different strands of cellulose form extended ribbon. Interchain hydrogen bonding allows multi-chain interactions, forms cable-like structure. So because of this complex nature of this structure, cellulose is more complex, and that beta linkage will give more strength to the cellulose. It's very difficult to hydrolyze as well as both enzymatic as well as non-enzymatic method. It's very difficult to break. That's why the fibers or plant material which is made up of cellulose is relatively high strength than the alpha linkage. Chitin is another class of complex carbohydrate. Exoskeletons of crustaceans, insects and spider and cell walls of fungi. These are all available source of chitin. Similar to cellulose, almost the same, but instead of glucose, N-acetyl glucosamine at C2 position available in case of OH from glucose unit. That is the difference between glucose as well as chitin, or cellulose as well as chitin. The beta-1,4 linked N-acetyl glucosamine units, cellulose strands are parallel, chitins can be parallel or antiparallel. Both way it is possible. Chitin, which is present here, the structure between two glucose unit, the second position having N-acetyl glucosamine, and it may be reorient themselves, will produce all beta-1,4 glycosidic linkage between two unit, will produce such a complex carbohydrate, namely chitin. The difference between cellulose and chitin, so simple. Cellulose is linked between two sugar unit via 1,4 beta linkage. In case of chitin, similarly 1,4 beta linkage, the second position having N-acetyl group, N-acetyl amino group, which is present in chitin, whereas hydroxy group present in case of cellulose. That is the only difference between these two polysaccharides. Glycoproteins, it may be two types. Glycosides, which is linked with proteins, either it may be linked via O or it may be linked via N. If it is nitrogen amino acid having the amino group, that nitrogen linked with glycosides to make a bond in, that is called N-linked glycopeptide. In case of amino acids having oxygen also, that oxygen makes bond with this glycoproteins will give O-linked glycoproteins. N-linked saccharides are attached via amide nitrogens of asparagine residues, whereas in case of O-linked saccharides are attached to the hydroxyl group of serine, threonine or hydroxylene, lysines. So these are all the possible to make glycoproteins. As an example of glycoprotein, where the 2 units or 3 units of sugar linked together apart from non-sugar part will give the glycoproteins. In case of O-linked glycoproteins, the function in many cases is to adapt extended conformations, and this extended conformation resembles Bristol brushes. Bristol brush structure extends functional domain up from membrane surface. So O-linked glycoproteins, which is present in our skins, which is responsible for the activities of our skins, in case of O-linked glycoproteins, which means that amino acids having the hydroxy group which is linked to the carbohydrate sugar portion will make the ether linkage, that is called O-linked glycoproteins. Which involves O-linked glycoproteins, that means the sugar having oxygen which is linked to protein, that protein is called glycoprotein. The glycoprotein, the linkage is via oxygen, which will give complex molecule which will be functionally, or it will carry out biochemical reaction in our body. Blood ABO antigens, structure of ABO blood group carbohydrates or represents the linkage to protein in this segregated forms, sphingolipid in the cell surface bound form, open square galnac, open diamond galactose, filled square fructose, filled diamond galnac, filled diamond sialic acid, etc. In case of N-linked glycoproteins, where the amino acid having the nitrogen or amino group which will link to the sugar portion will make N-linked glycoproteins. Oligosaccharides can alter the chemical and physical properties of proteins. Compared to monosaccharide, oligosaccharide is more than 3 to 10 units of monosaccharide which can link together, and it will link to further to proteins that will make entire function of the proteins. And also oligosaccharides can stabilize protein conformations and protect against proteolysis. Proteolysis means protein hydrolysis, that can be prevented by addition of oligosaccharides. And cleavage of monosaccharide unit from N-linked glycoproteins in blood targets them for degradations in the liver. So this is also another important role of N-linked glycoproteins involved in targeting protein to specific subcellular compartment. So these are all the major class of N-linked glycoproteins which will involve in the function animal as well as in human being. N-linked glycoproteins, similar like glucose, if hydroxy group present, that is linked with the protein, that is called O-linked glycoprotein. N-linked means the sugar having amino group, that amino group which is linked with protein molecules, that is called N-linked glycoproteins, which is again very important role in place of functional molecule as well as play in the biochemical reactions in the animals as well as plants. So students, let us now summarize what we have learned in these modules. So we have learned basics of carbohydrate, that means what are carbohydrates and basic simplest carbohydrates, oligosaccharides and polysaccharides, and also we have further studied monosaccharides and its properties and applications, oligosaccharides and its properties, applications also we studied, and in case of polysaccharides some of the well-known examples and its structure we have studied, and examples of polysaccharide like 6 examples we studied fully, and what is the orientation and the structure and the function of that particular polysaccharides, and application of polysaccharide we have studied in details. Apart from that, glycoproteins and its function, especially O-linked glycoproteins or N-linked glycoproteins, that we studied in details.